Welcome everyone to Chapter 12, The Molecular Basis of Genetic Disease. And this will be in two parts, and this is part one. We'll start with disorders of the hemoglobin. Hemoglobinopathies exemplify molecular diseases where genetic alterations actually lead to disorders. By examining hemoglobin disorders, we can appreciate how understanding gene variants at a molecular level is critical in advancing therapeutic options. One of the first examples would be sickle cell disease, which was the first genetic disease studied at a molecular level. So there are a few mechanisms by which pathogenic variants result in disease in the patient. Loss of function occurs when a protein is either reduced or completely non-functional, and this is often due to deletions, nonsense mutations, or frame shift mutations in the gene. Gain of function involves excess or abnormal activity of a protein, and this is typically caused by mutations that increase activity or confer a new property. Novel function refers to the development of an entirely new function for a protein, which can lead to disease. And misexpression happens when a gene is expressed at the wrong time or in the wrong location, so in the wrong tissue entirely, thus disrupting normal biological processes. So a clear understanding of these mechanisms is actually quite important for developing targeted treatments for genetic disorders. By pinpointing whether the issue is due to loss, gain, or novel function, or misexpression, therapies can better be tailored to address the underlying cause. So let's start by discussing loss of function variants or mutations. So pathogenic variants can arise from deletions, substitutions, insertions, or rearrangements. And these changes may impact coding sequences or regulatory elements, altering gene function or expression. An example would be alpha thalassemia, which results from a deletion of one or more alpha globin genes, leading to reduced gene dosage and anemia. Turner syndrome is a chromosomal aneuploidy characterized by the loss of one sex chromosome, resulting in the developmental and reproductive abnormalities in the carrier, and cancer, which is acquired somatic mutations in tumor suppressor cells or genes, such as the RB1 gene in retinoblastoma, which lead to uncontrolled cell growth. The severity of a genetic disorder often correlates with the extent of functional loss. Partial retention of function can reduce disease severity, while complete loss typically results in more severe outcomes in the patients. Gain of function variants or mutations, on the other hand, enhance the normal activity or production of a protein, which can often lead to disease. There are different types. So there's increased protein production, which is seen in conditions such as Down syndrome due to trisomy 21 and Charcot-Marie-Tooth disease type 1A caused by gene duplications. There's also enhanced protein function, and where some variants alter protein behavior, such as the hemoglobin Kempsey variant, which keeps hemoglobin locked in a high oxygen affinity and disrupts oxygen release to tissues. So understanding gain of function variants is actually quite important for developing tailored treatments as well. These approaches often differ significantly from those used for loss of function variants, such as they aim to reduce or modulate the excess activity rather than restore functionality. We're going to focus on the globin genes to start with because they do provide a clear example of how different genetic variants can disrupt protein formation and lead to distinct diseases such as thalassemias. So there are different steps in protein production affected by variants. We have transcription, where variants can reduce or completely prevent the production of messenger RNA, leading to decreased protein synthesis. In translation, mutations can produce non-functional or unstable mRNA, hindering the translation process. We have folding, where abnormal protein folding results in unstable proteins that are often targeted for degradation, reducing functional protein levels. Or in assembly, where errors during the assembly of protein subunits can impair the overall structure and function of the final protein complex. So understanding these disruptions and when they occur is critical for linking genetic changes to clinical outcomes and for the developing target therapies. So variants affecting the globin genes provide a clear example of how disruptions in each stage of protein production can lead to a range of clinical diseases, particularly in hemoglobin formation. So as I mentioned, each stage in protein production, from transcription to functional activity, can be disrupted by genetic variants or mutations. These disruptions are reflected in a variety of diseases, and these are outlined in Table 12.1. But I just wanted to go through some of them with you as an example. Um, so some of them would be thalassemias. We have disruptions in transcription or translation. In protein folding, we have examples in HB Hammersmith. Localization, which is misplacement of proteins. An example is familial hypercholesterolemia, which is high cholesterol that is inherited due to mutations in various genes. And disorders in protein function, which could be structural but dysfunctional proteins are produced. Example would be HB Kemsey disease. In the next few slides, we'll be discussing the relationship between genotype, which is the genetic makeup of a disease, and phenotype, which is the outcome of the disease disorder and genetic disease. So variants that disrupt the highly conserved structure of hemoglobin often lead to disease, as these changes impair hemoglobin's ability to efficiently carry oxygen throughout the body. So examples would be that mutations that affect the critical amino acid in the globin chains can produce unstable or dysfunctional hemoglobin molecules. These alterations result in conditions known as hemoglobinopathies, such as sickle cell anemia or other structurally abnormal hemoglobins. So understanding these structural disruptions help in diagnosing and developing targeted therapies for hemoglobin-related disorders. So the relationship between genotype and phenotype in genetic disorders or diseases is influenced by factors such as allelic and locus heterogeneity, as well as modifier genes, which can alter the way a disease is expressed clinically in patients. So what are the different genetic heterogeneity? There we have allelic heterogeneity. This is where different mutations within the same gene can result in varying disease phenotypes. We have locus heterogeneity. This is where mutations in distinct genes can lead to a similar clinical presentation, highlighting overlapping biological pathways. And we have modifier genes. So additional genes can modulate the severity or characteristic of a primary genetic disorder. Examples would be the alpha globin and beta globin genes, illustrating these principles. Variants in these genes contribute to a diverse clinical outcome, such as thalassemias or hemoglobinopathies. So this really depends on the specific mutation and the influence of modifier genes. These concepts are critical for understanding the complexity of genetic disorders. So we'll start with allelic heterogeneity. So this is where different mutations, as I mentioned, such as deletions or variants occurring in the same gene locus or location, can lead to the same disease, though the severity of the disease may vary depending on the specific mutation. Example would be beta thalassemia. This is where variants in the beta globin gene result in differing severities of beta thalassemia, ranging from mild to severe based on the extent of beta globin production impairments. HB-Chem-C is a perfect example. This is a specific beta globin allele causes hemoglobin to remain in a high oxygen affinity state, reducing oxygen release to tissues and leading to polycythemia. Locus heterogeneity, on the other hand, is when different mutations, deletions, or variants in distinct genes can lead to a similar clinical phenotype, demonstrating the convergence of diverse genetic pathways on shared biological outcomes. Thalassemia is a prime example of locus heterogeneity. It can arise from mutations in either the alpha globin or beta globin genes. Yet, the clinical presentation often overlaps and involves anemia and related complications in the patient. Submodifier genes are additional genes that influence the expression or severity of a primary genetic disorder. They can either exacerbate or mitigate the disease phenotype. So an example would be beta thalassemia. When co-inherited with an alpha globin gene deletion, this can reduce the severity of beta thalassemia symptoms by balancing the globin chain ratio. So it actually can make the clinical burden a little bit better on the patient. So modifier genes play a significant role in explaining variability in disease severity in patients, even when the primary mutation is well understood. This highlights the complexity of genetic disorders and the importance of considering modifier effects in both the diagnosis and the treatment strategies for the patient. So hemoglobinopathies are a critical area of study, not only because of their prevalence being the most common monogenic disease globally, but also because they have greatly contributed to our understanding of genetic diseases and gene structure. Hemoglobin is the oxygen-carrying molecule in red blood cells. It consists of four subunits, two alpha globin and two beta globin chains. Each subunit includes a globin protein and an iron-containing heme group, which is essential for oxygen binding and transport across the body. The primary adult hemoglobin, hemoglobin A, or HbA for short, has a structure denoted as alpha 2 beta 2. Its tertiary structure is highly conserved to ensure proper function. However, certain regions of the molecule are particularly sensitive, and variants affecting these areas can disrupt its function, leading to diseases such as the disease of the globin. The globin genes are organized into two main clusters based on their evolutionary relationship. Alpha-like genes are located on chromosome 16, and this cluster includes alpha-1 and alpha-2 genes. The beta-like genes are found on chromosome 11, and this cluster includes the beta and gamma and other related genes. Different types of hemoglobin are actually expressed during various stages of development of the human being to meet changing oxygen needs. For example, HbF, which is the fetal hemoglobin, actually dominates during fetal development with a higher oxygen affinity for efficient transfer from maternal blood. Globin switching refers to the developmental regulation of globin gene expression, where different globin genes are activated, so turned off or turned on, at specific stages of development. The genes within the alpha and beta clusters are arranged in the order of their expression during embryonic, fetal, and adult life. So during the 3rd to 8th week of gestation, embryonic globins are synthesized in the yolk sac. By the 5th week of gestation, erythropoiesis transitions to the fetal liver, marking a shift in globin expression. HbF, which is composed of different gene combinations, predominates during fetal life. At birth, it constitutes approximately 70% of total hemoglobin, but this slowly decreases to only a few percent in adults as it's replaced by adult hemoglobin, which is HbA. The expression of beta globin gene cluster is tightly controlled by the locus control region, or LCR, which is a regulatory element located upstream of the beta globin cluster. The LCR is critical for helping the developmental transition from embryonic globin to the beta globin. So the LCR contains DNase 1 hypersensitive sites which promote an open chromatin structure, allowing transcription factors to access the globin genes. It forms an active chromatin hub, enabling interaction or communication between the LCR and specific globin genes within the cluster. This mechanism supports the activation of globin genes during different stages of development. The LCR is actually really important because it helps ensure proper globin gene expression and so it remains turned on and maintains the developmental regulation of hemoglobin synthesis. The gene dosage for globin gene varies significantly between the alpha and beta clusters. So each diploid genome contains 4 alpha globin genes, and this kind of distributes the impact of mutations. So if you've got a mutation in one of the 4, 25% of the hemoglobin is affected. If you've got in 2 genes, that's 50%, and so forth and so forth. But you've only got 2 beta globin genes, so a single mutation actually affects 50% of the globin gene chains, compared to only 25% in an alpha globin chain for a single mutation. So beta globin variants actually have no prenatal effect, so the fetus is not affected. The symptoms usually emerge postnatally as beta globin becomes a primary globin. But for alpha globin variants, these lead to disease during both fetal and postnatal life. This is due to the alpha globin's exclusive role in hemoglobin synthesis starting from 6 weeks post-conception. Understanding these differences in gene dosage and timing highlights the distinct impact of alpha and beta globin gene mutations on these diseases.